Hello, welcome to the Stem Cell Biology lecture series of the University of Vienna. My name is Noelia Urban. I'm from IMBA and I will be sharing with you the lecture on adult neural stem cells, which I hope you will find interesting. Let me just put this in full screen. Perfect. So let's get started. The way I structure the lecture is that I will give you a brief intro on adult stem cells in general. And then I will go a little bit deeper into adult neurogenesis. So not only the neural stem cells, but how it was found, where can we find this process, and what's its function. And then I will move to the main part, which will be on adult neural stem cells and their very special state of quiescence, how that's regulated, and how heterogeneous this state is. I will also touch a little bit on the topic of human adult neurogenesis in the end and how novel methods can help us know a little bit more about this in humans. For the last two parts, I will also intermingle some of the research that we are doing in the lab that is relevant to these parts so that you see which type of things are being done at the moment in the terms of research for this topic. Okay, so in a very general sense of stem cells, you've been hearing about them a lot, so I think this will be nothing new, but there are different types of it if you think of their potential and also when during the lifetime of the organism they are found. You have in the very early stages in the morula these totipotent stem cells, which will give rise to any tissue. So they can generate the fetus itself, but also the extraembryonic tissue. These cells then start differentiation and differentiating and acquiring different fates. And by the stage of the blastocyst, they are already not totipotent anymore because different cell types will be able or not to give rise to extraembryonic tissue or the fetus. And the most famous of these blastocyst stage are actually the ones here in what is called the inner cell mass, which are the pluripotent stem cells, which can give rise to the whole organism, but not the extraembryonic tissue. These inner cell mass cells then will undergo a lot of differentiation steps, gastrulation, fate determination. They will give rise to the three germ layers. And in the tissues that they will keep generating, there will still be some stem cells that in this case we call multipotent and in some cases even unipotent. So they will be residing in those tissues and continue generating those tissues during development and also in adulthood. So during adult stages, we still have stem cells in our tissues that help us keep the homeostasis of that tissue. These stem cells have a few characteristics that distinguish them from the embryonic ones. One of them is that they don't proliferate as much, they are mostly quiescent, and we will go back to this concept a little bit later. Another one is that they tend to divide asymmetrically or be very stereotypical in their divisions, even though this is also not generalizable for all of them. And the other one is that they tend to not have a specific function when they are stem cells, but the function comes from the differentiated cells. So they just provide a source of a range of different cells for the tissue that then will have this function. These stem cells, as I said, are in many different tissues and they are very well known. They have different characteristics, different environments. For instance, the ones in the skin are constantly renewing our skin or our gut, also same thing. And in some others, there's a little bit less obvious, like in the skeletal muscle, they might be a little bit less active and only upon injury they get activated and then regenerate the tissue. And here it's already listed the brain, but actually for a long time it was a bit controversial whether in the brain there will be this ability of having stem cells and generating new neurons. And this came from a very strong dogma initiated by Santiago Ramón y Cajal, which is considered the father of neuroscience, of modern neuroscience, because he wrote that once the development was ended, the founts of growth and regeneration of the axons and dendrites dried up irrevocably. In the adult centers, the nerve paths are something fixed, ended, and immutable. Everything may die, nothing may be regenerated. So you can see he had this very flourishly style of writing. But even though most people stops here and takes it as the time that he got it wrong, he actually wrote a little bit farther. And his next sentence is that it is for us, the science of the future, to change, if possible, this harsh decree. And actually, a few years after he wrote this, so in the, well, quite a few years, in the 60s, um, Altman and Das did find some new neurons that were generated in the rat brain, in the hippocampus. So this will be the dendrite of the hippocampus. And they found them by administering thymidine analogs to these mice, in this case radioactively labeled. And they could see that new neurons, because they had proliferated and they had incorporated this thymidine analog, could be found in the dentate gyrus. They also found it in another niche in the subventricular zone. But funny enough, no one paid much attention to these findings, and they were mostly dismissed by the neuroscience community. And it was not only until Fernando Nottebohm in the 80s found that the songbirds were also incorporating new neurons every year in order to learn all these new songs that you can hear, for instance, now in spring, that people started to look back at those works by Altman and Das in rodents and start realizing that indeed it was possible that new neurons were being generated here. But I would say the boom of coming back to this adult neurogenic concept in the brain didn't come until the 2000s. So it's roughly 20 years since we are really focusing on this and getting much more evidence of this happening. Adult neurogenesis in the adult rodent brain is now very well established. It happens throughout life and it happens in different niches. And more of these niches are being found. But the two main ones are the dentate gyrus in the hippocampus and the subventricular zone in the lateral ventricles. Yeah, so just to say that even though there are many niches that are being discovered, and these two are very clear, this is not a process that happens throughout the brain. And the numbers of new neurons, if you make it relative to the number of older neurons that are there, are relatively low, but they still play a very important function. So, for instance, the ones in the subventricular zone, let me show you with the pointer, which is here. So the stem cells will be located here and they will generate neuroblasts, well, intermediate progenitor cells, and the neuroblasts that then will migrate through a stream called the rostral migratory stream to the olfactory bulb of the mice, where they integrate into the existing circuit. And this will be mostly interneurons, different types, depending on where in the subventricular zone you are. And they will play a function in the olfaction of the mice, which is very important for them. The other main niche is in the hippocampus, in the dentate gyrus, where in the subgranular zone, so just below the layer of neurons, there will be some stem cells that also generate intermediate precursor cells, neuroblasts, and then mature neurons that integrate in this circuitry. And I will mostly focus on this niche because it's the one that also has been found to be active in humans, and we'll go back to this later. And it's also the one that we are studying in the lab. But I will also give you some examples and some literature regarding the other main niche, the subventricular zone. So focusing now on the dentate gyrus, as I told you, there's these stem cells that will activate and then generate intermediate precursors, late intermediate precursors, neuroblasts, and they will eventually differentiate. This process is very similar to what happens during development for the generation of the dentate gyrus, of the main neurons that are there. So we can find markers that are very similar, and also the process and timings are relatively similar. And these neurons, what I find absolutely remarkable is that even though there's not an active developmental environment between when they mature, they can still fully integrate into the functioning circuitry of the hippocampus. So this is now again the dentate gyrus here. So the neural stem cells will be here and then will generate neurons that in the case of the dentate gyrus, they always stay locally. So they never migrate away, they stay in the dentate gyrus. And here you can see one of these cells, and it will fully integrate into the circuitry of the hippocampus. So I'm showing here the trisynaptic circuitries, which is one of the most important circuits and most studied in the hippocampus, where cells from the entorhinal cortex will send inputs to these granule cells in the dentate gyrus, of which some of them will be newborn cells. And then these cells will send their axons to the CA3 region of the dentate gyrus, where there's these pyramidal neurons that in turn send their axons to the CA1 pyramidal neurons, which send the information back to the entorhinal cortex after this processing. There are many other connections of the dentate gyrus, some of which I will tell you about a bit later. But what do these neurons do then in this circuit? So one of the most studied things that they do is to give you the ability of having plasticity in your spatial learning. And here is an example of this. So this will be a behavioral test in which they are trying to see if the mice can distinguish between one context, so that will be context A, and one very similar context, which is context B. And this is because mice without adult neurogenesis are able to distinguish things that are very different, but not things that are very similar. So one example that is normally put forward is that they will be able to distinguish between an image of an apple and a pear, but if you put two similar apples, then they will really struggle. And the reason for this is that the new neurons help the old neurons to be much more restrictive in how they activate in response to cues and to things that are similar. So for instance, the example that they put here is that if you don't have neurogenesis, the engram or the neurons that you recall when you see this new context will be very similar to the first context. So you won't be able to really tell whether they are different. However, with neurogenesis, the new neurons are very highly activable. So you will have many of them that are active, and that will help inhibit the older neurons because it turns out these very activable young neurons are very well integrated in the system in terms that they can activate interneurons very efficiently, but they still don't receive the inhibitory input from the interneurons so well. So they are really immune to these inhibitory signals, which they are activating. But since they are increasing this inhibition and the older neurons are very much inhibited by this inhibitory input, then it will restrict a lot the number of older neurons that are activated. And then these mice then can distinguish between these two different contexts. Sorry. Another example that I also find very interesting is in the Morris water maze, in which, just in case you are not familiar, the mice are put in a swimming pool. It's normally a circular swimming pool in which the water has a dye, so it's like white paint. It looks like milk, so they cannot see below. And there is only one platform where they can go and rest there. Normally, mice don't like to swim so much. That depends a bit on the strain. But if you put them there, they will really try to find the platform. And what you normally do is to put some cues for them to really be able to learn where this platform is. So it turns out with and without adult neurogenesis, the mice can very well learn where that platform is. But there is one assay in which once they have learned, you move the platform from its original location and then test whether the mice are able to relearn that the platform is no longer there and they have to find it somewhere else. So with adult neurogenesis or normal mice can do this very efficiently as well. So they will initially try to find the platform where it originally was, but then they very rapidly learn that it's not there and then can locate the new location of the platform. If you don't have neurogenesis, as I said, the mice will have no problem in finding initially the platform, but when it changes location, they won't be able to learn that it's not there anymore. So somehow they became more stubborn. And this is something that often is associated also with old age and this lack of plasticity. So always thinking that things should be as as they were. But of course, human to mouse direct applications and these type of things are a little bit far-fetched, but I find this very interesting. And these two examples are about this spatial memory and capacity of spatial navigation, but and this is mostly what the dorsal hippocampus does. But there's also a very important connection in the circuitry of the ventral hypothalamus to other parts of the brain, like the medial prefrontal cortex, the nucleus accumbens, the basolateral amygdala, and the hypothalamus that are involved in much more um, in much different things, more related to fear, to reward, to pleasure, to anxiety, to stress. And this is also something in which adult neurogenesis has been highly involved. And I really recommend you to check some of the reviews if you're interested in this. But for instance, it's also very important for stress regulation. So with high neurogenesis, it's supposed that this activity of these newborn cells will really help you minimize the amount of activation that you have of these regions that will give you a lot of anxiety and fear responses. And also when the stress is not there anymore, not only you will have had a little bit lower stress, but then you are able to come back to a no stress situation. While if you don't have this input of new neurons, this contribution of them, initially your stress will already be much higher and you won't have that much of an ability to go back to your baseline levels of stress. Yeah, and then moving on a little bit. So I really recommend you to go for further reviews because it's very fascinating and we are still learning about what adult neurogenesis do and also about what happens in humans, which again, we will get to back to that a little bit later. So one characteristic that is different to what happens during development of these adult neural stem cells is that they are not engaging in this neurogenic process continuously, but they are in what we call a quiescent state. So basically, they are just sort of sitting there, listening to their environment and deciding whether they need to go on and generate neurons or not. And I would say over 95% or even more of the neural stem cells in the dentate gyrus at any given point are in this quiescent state. This quiescence is very important, not only because, of course, if you don't activate, you don't produce neurons, but also you can see here with these arrows the neural stem cells are not very good at self-renewing, actually, which means that the more you activate them, the more you lose them through producing neurogenesis. So the active neural stem cells might not be able to give rise to two or even to one new neural stem cells when it divides, which means the more they activate, the more they deplete. So it's very important to maintain a good control of this quiescence to activation transition and also to the active to quiescent transition in order to maintain a good number of new neurons being generated, but not exhausting the neural stem cell pool. And this, I mean, going back to different tissues, this is different in different tissues. So as I told you, skin and gut are constantly renewing. So quiescence is not a prominent feature of them. They seem to be in an actively, like constant on state. While in other tissues like the skeletal muscle that I also mentioned, the cells are not doing anything in normal homeostasis, but only when there's an injury. There's also mixed systems like the blood, where there are a subset of very quiescent neural stem cells that act as some sort of reserve and are only activated when there's an hemorrhage. But there's also a pool of very active neural stem cells that are constantly renewing our blood. And the brain will lay in the more towards the quiescent state. So quiescence is a very important feature in this cell. So what is this quiescent state? So it's basically an extra cell cycle phase. So it's a G0 state, but that is fully reversible. And that distinguishes it from things like senescence, which happens during aging or upon stress, which is an irreversible G0, where the cells also will have a very particular secretory phenotype that will affect their environment. And it's also different to terminal differentiation, where the cells go on and acquire a different fate. So they really differentiate into a terminal cell that then is not able to go back to a proliferative state and to this stemness. So it's basically maintaining stemness, but without dividing, always retaining this capacity to redivide. But this is not the only characteristic, of course, of quiescence. Quiescent cells in many tissues share other characteristics, so they tend to handle their transcription and translation a little bit different. They have a lot of intron retention. They have different heterochromatin marks also. They retain a lot of mRNAs in granules. They increase their levels of autophagy also and decrease mTOR signaling. And all this gives them a very special state that is supposed to protect them also from damage. For instance, reactive oxygen species. In the adult neural stem cells, there's a lot of different pathways that have been involved in their regulation. So this is just a small summary. Again, if you're interested, please go to the literature and find these reviews. But the regulation is very active actually. So this might be a little bit counterintuitive because they are called quiescent, but that's just because they are not in the cell cycle, but they are very actively listening and sensing their environment. So, for instance, the other cells will talk to them through delta notch signaling. There will be plenty of contacts also with the extracellular matrix and also with signaling components that are all around and secreted by the niche cells and also by themselves. Yeah. So this regulation is very, very important. So this is one of the things that us and many other groups are interested in knowing what is controlling this quiescence to activation transition. And over the next few slides, we will be having a look at how is it acquired and how is it maintained and regulated. So first of all, with how does this happen in the first place? So the classical view of how neural stem cells develop over time is that you will first have neuroepithelial cells. You might have seen this already, which are highly proliferative and mostly divide symmetrically to increase the pool of neuroepithelial cells, which then will make a switch to be dividing asymmetrically, which means that they will generate one more of themselves, but also start generating neurons. And in that state, they are called radial glia. These radial glia during development will generate all the neuronal types. In this case, the example is for the cortical plate, for the cortex. And once they have generated all the neurons, they will have a gliogenic switch and they will start to generate glia, both astrocytes and oligodendrocytes. They will also then generate ependymal cells later on around postnatal, early postnatal. And this is the time where it was thought that they also get converted into what are called B cells, which are the stem cells in the subventricular zone niche in the adult. But this happened to not be quite like this because there were two parallel manuscripts published in 2015 that showed through different and very nice and elegant ways that actually the neural stem cells were stopping dividing, so acquiring quiescence already during embryonic development. So what they found is that there was a subset of these radial glial cells that will stop dividing around embryonic day E14 to E15. They will start to express high levels of cell cycle inhibitors and really stop cycling, so they will not go on to generate astrocytes or oligodendrocytes or ependymal cells. And then it will be these cells that stop here that are actually the adult stem cells of later that will then get reactivated much later on. And this is thought to help them not lose their neurogenic potential. Because as I told you, there's a gliogenic switch happening late in embryonic development and around birth. That means that the radial glia is not able to generate neurons, or it does not generate neurons anymore, but it generates glia. So by inducing quiescence before that switch, it's thought that that's the mechanism by which these adult neural stem cells can still produce neurons in the adult after such a long time. But this actually is not the same in the other niche. And this is something very interesting, that it's often similar, but a little bit different. So in the dentate gyrus, what happens is that there's a very gradual entry into quiescence. So the same type of cells, without a separation between the ones that will become adult and the ones that generate the bulk of the neurons of the dentate gyrus, will be undergoing this generation of neurons, and then gradually, slowly, during the first couple of postnatal weeks, they will become um, quiescent, and they will also acquire their location in what is called the subgranular zone, so below the layer of neurons, which I find is quite remarkable. Okay, so now I will tell you a little bit of what can we do to study these cells in the adult already. So one thing that has been very much used and very, very useful is genetic labeling and lineage tracing. So what we can do is to use genetics of mice. So for instance, in this case, I'm showing you one image of the hippocampus of a mouse which has a GLAS CRE32 allele. So GLAS is expressed in astrocytes and in stem cells. And CRE32 is a CRE recombinase that is activated when tamoxifen is added. So it goes to the nucleus and can delete genes or LogSP cassettes in there when you add tamoxifen only, which means that we can very nicely temporarily control the activation of any expression or like whatever we want. In this case, it's a reporter, a ROSA stop YFP. So there will be a LogSP cassette which has which surrounds a stop codon just before the YFP protein, which means that only in the cells that express GLAST and only when we administer tamoxifen, which in this case was at 8 weeks of age, the CRE will be active and will delete this stop cassette, and then the cells that were GLAST at that time will become green forever. So you can then follow their fate over time. And you can see that astrocytes, because GLAST also labels astrocytes, are everywhere. So it will be all these little green cells all around. But there's something special happening in the dentate gyrus, which is in the subgranular zone. You have this beautiful layering of neural stem cells. And then we can also couple this to the deletion of particular genes. So you can imagine that instead of having the LogSP surrounding this stop codon, you can have it surrounding the whatever is your gene of interest. And then you can have a conditional knockout with temporal control of when you start to have the deletion. And one other tool that we have that is very useful for molecular characterization, because you saw how not very abundant these stem cells are, is that we can isolate the neural stem cells from the mouse brain. We culture them in vitro. They can be expanded for many passages, actually. They are relatively easy to amplify. And we have one little trick, which was first described by the lab of Elena Mira, in which we add BMP4 to these neural stem cells, and they will acquire a reversible quiescent state. And in this state, they will upregulate a lot of astroglial features, which are characteristic of these cells in vivo as well. And they will, of course, withdraw from the cell cycle. And this needs to be reversible, and it is. So you can see here that proliferation, marked by the proliferation marker Ki67, really drops dramatically when we add this BMP. But when we remove it, the cells then will go back in 2 days to the same level that they had before. And they still can differentiate, so they really are indistinguishable from their initial counterparts before going through the quiescent state. And with this, then we can really start to study in much more detail how specific signals or specific perturbations affect these transitions. So one thing that we learned, for instance, with this is that there was a specific transcription factor called ASL1 that we had observed that was expressed in the active neural stem cells, but also in the progenitors. And this is a transcription factor that has been shown to be pro-neurogenic. So if you overexpress ASL1 even in an embryonic stem cell or in a fibroblast, you can convert it into a neuron. It's a very, very potent pro-neurogenic factor. And we found it was expressed already in the active neural stem cells. So we decided to delete it with this same technique that I told you. So in the GLAST expressing cells, only in the adult animals. And what we saw is that deletion of ASL1 not only impaired the generation of neurons, but also the activation of these quiescent neural stem cells. And since they did not activate, they also did not deplete. So we saw that while normally, because of this activation coupled depletion in older mice, you start to see a decrease in the number of neural stem cells. In these mice which didn't have ASL1, this did not happen anymore. And yeah, so this transition will not happen. So that told us that ASL1 was very important for the activation, actually crucial for the activation of these cells. And we started to evaluate or to investigate what could be regulating ASL1 because we thought its levels will also be very important for determining how much these cells activate. And we found one very important regulator, which is HUVE1. HUVE1 is an E3 ubiquitin ligase that directly targets ASL1 for proteasomal degradation. We found that ASL1 levels at the mRNA level were not different between quiescent and active cells, but the protein was. It was never found in quiescent cells and only in the ones that were active. And when we use this same technique I told you of GLASS Creativity 2, and in this case combined with deletion of HUVE1, we saw that there was little change to the quiescence to activation transition of these cells. But when HUVE1 was not there, ASL1 levels were much higher and the cells were not able to rapidly degrade ASL1 to be able to return to a quiescent state. And that actually led to a loss of neurogenesis in these cells. So they proliferated even less because only the very deep quiescent ones were able to remain. And there was also a block on neurogenesis, but again, you can check the paper for further details. But one very interesting, let's say, side observation that we made that was that by not allowing these active neurostem cells to return to quiescence, we also lost most of the proliferation, as I told you. And that told us that there was actually two different types of neural stem cells here. So we had what we call dormant, which was these ones that were very difficult to activate and were hardly affected by HUVE1, and those that had recently returned to activation, which was not possible without HUVE, so HUVE1 is required for this. And we call them resting because they were much more likely to go back to an active state. And that meant that also they were the main contributors to neurogenesis. So most of the neuroblasts and neurons were coming from these resting cells and obviously not from these dormant ones that were barely activating. And that gave us the first hint of some heterogeneity between the neural stem cells in the dentate gyrus. So we also saw that these dynamics of dormant, active, resting, and the involvement of HUVE1 and ASL1 will change during aging. So in aging, the effect of HUVE1 got even bigger. So it seemed that it was a little bit more expressed, but we think it was also more active, meaning that fewer dormant neural stem cells are able to activate, but more importantly, many more of the stem cells that activate then go back to quiescence instead of continuing generating neurons and depleting. And we believe that this is behind the further preservation of the pool when you are older. So you start to lose your stem cells rapidly when you're young, at least in mice, But the slope declines very much in older animals. And we think this is related to these changing dynamics between the transitions of states. And this is again summarized here where ASL1 protein really decreases over time. And we think this HUVA1 activity is what keeps this change that helps to maintain the stem cells over time. Okay, so as I told you, we started to see this difference. So maybe this sort of simple transition between quiescent and active that we have drawn here was not really true. And indeed, then many other groups started to see that by genetic lineage tracing using different promoters, they saw different behaviors of stem cells, which means that neural stem cells are not homogeneous, but they come in many different flavors. And this is something that we are still trying to understand in the field. And there's two main sources for this heterogeneity. One of them has been mostly proposed through single cell sequencing experiments, where you can run a pseudo-time and then order neural stem cells depending on how quiescent or active they are. So there will be deep quiescent cells and then others that are almost active. And then there's a lot of heterogeneity in the stem cell population between these, meaning that it's not an on-off switch, but rather something much more gradual. And as I just mentioned, also there were different populations of neural stem cells that were identified that had not only different depths of quiescence, but also different ability to generate neurons, glia, and self-renew. And the, I would say, funny thing is that there's no evidence in the single cell sequencing data of the existence of these different populations. This can have many reasons, but we think also that we need a better model to integrate these views. And what we propose is that instead of this very linear transition that has been proposed between deep quiescence and activation, what happens is that stem cells are in a very dynamically heterogeneous state in which they will constantly move, meaning that you can have a very different profile of these neural stem cells at every level of depth of quiescence. So they are really not there's not a stereotypical way to go from quiescence to activation. And we believe that this is very important for their function because it gives them the ability to be different, meaning that they can respond to many different cues as a population. And that also allows them to avoid stem cell exhaustion. Because if you think that you might be receiving a repeated stimuli that activate your stem cells, you don't want all of them to activate because they don't self-renew very well, so then you will lose them. While if you have only specific subsets that activate at specific time points, then you will always have some that respond, but others that did not and are there for the next time that you are going to receive that stimuli. And what we want to know in our lab and actually in many other labs is how this behavior and this what we call cloud of states of the neural stem cells changing with aging when you change conditions like diet or are exposed to stress, and also how this is regulated by the niche signals. And yeah, and here I want to introduce you another big thing in the field, which is that neural stem cells are extremely similar to astrocytes, to these glial cells. And what we think and what many have observed is that many of the markers of neural stem cells, especially of quiescent neural stem cells, are really indistinguishable from those of astrocytes, even though their morphology and location within the niche are very different. So here I draw now an astrocyte, which you can see that has nothing to do morphologically with a neural stem cell, but the markers and also when we do transcriptomics are extremely similar. It's very difficult to disentangle these two states. So this is something that in the lab, Mara, who was a lab member in the past, and now Alejandro, who is a PhD student, is trying to tackle. So first, how heterogeneous these cells are, but of course, first to test heterogeneity, you need to distinguish them from these very similar astrocytes. And I'm just going to show you how this similarity actually looks by showing you two of these markers, which are SOX2, which is a transcription factor that is expressed by astrocytes all the way to the intermediate precursor cells, and also GFAP, which is a very common marker for astrocytes, but that is also expressed by both quiescent and active neural stem cells. So now we see like the real thing. So this will be the subgranular zone in here. This is the granule cell layer, so all the neurons will be here. And this is the molecular layer, so the axons from the entorhinal cortex will come here and contact these neurons. And this is the illus, which is in between the two blades of granule cells of the dentate gyrus. So in here with the SOX2, which is this nuclear marker, and the GFAP, which labels the projections, you can easily, for a trained eye, distinguish astrocytes, like for instance these two here, and also neural stem cells. So they also have their nuclei labeled with SOX2, and they have this very characteristic single projection that goes through the granular layer, reaching to the molecular layer. And you can even see that there's the intermediate precursor cells, which lost already the projections, the GFAP positive projections, but they still retain the SOX2 positiveness. Okay, so then Alejandro thought that he could use something like a lunatic fringe. So let me see, because there was another slide here, which I think, okay, it's not there. Yeah, so there are some markers which are more specific to neural stem cells, like nesting and lunatic fringe. So lunatic fringe is what I'm showing you here, that you can see that it's expressed by some astrocytes, but not all. But it's also expressed by the IPCs and many other cells, which are the progeny of the neural stem cells. So yeah, so here's the slide that I was looking for before. So there was a paper relatively recently that described that this marker, ALDH1 like 1, was exclusively expressed by astrocytes and not by stem cells in this niche in the dentate gyrus. And this we found very interesting because that will mean that we have a way of sorting them differently, and then see what's really the difference between these cells, or at least to have a pure population of neural stem cells and their progeny without having to worry to have contaminating astrocytes in here. So what we did was to get mice in which the Cre recombinase that I told you that is tamoxifen dependent is driven by ALDH1 like 1, meaning that we will be labeling, in theory, only the astrocytes and not the stem cells. And here I can show you again this same mice in which we were quite disappointed to see that actually only some astrocytes, you can see this one, yes, but this was no, was labeled by this transgenic line, but also some stem cells. So you can see this one no, but this one yes, and even some intermediate progenitor cells. So we cannot trust this marker either for this separation, and is again another, yeah, no, if you put all the markers together, it looks beautiful, but not very useful actually. But it's another demonstration of the high similarity between these two cell types. So we decided to look also to another marker, which will be nestin, which is also not supposed to be expressed by astrocytes, but it is expressed by quiescent and active neural stem cells, as well as some of their progeny. And actually this goes, if you use a GFP reporter, the GFP expression will also linger a little bit into the neuroblasts. So I'm showing you here again a SOX2 and GFAP staining. You can see this beautiful neural stem cell, SOX2 positive, with the projection here, which is GFAP positive. You can also see many astrocytes around. And this is a nesting GFP mice. So in this case, it's not a Cre technology, but simply all the cells expressing nesting will be also expressing GFP. So it's a direct transgene. And you can see that this very nicely marks our stem cells, but also some other cells in the niche, which we knew that this marker is also not specific of the neurogenic lineage, but also expressed by oligodendrocytes and endothelial cells. But what we found was good is that those astrocytes, which are SOX2 and GFAP, are not green, meaning that at least we can then, with this marker, distinguish these two cell types. So we did single cell sequencing of this. So basically, we dissected, and when I say we, it's Mara and Alejandro, the dentate gyrus, then made a single cell suspension and ran through the through our transcriptomics facility with 10x. And we could retrieve nicely endothelial cells, oligodendrocyte cells, and all the neurogenic lineage, so what we were expecting. But quite unexpectedly, because we counted also these sections, and we saw that there was a very small percentage of astrocytes, and we had a good percentage of neural stem cells. What we saw was that we were retrieving very few neural stem cells and a lot of what was annotated as juvenile and adult astrocytes. So this was very shocking. And then we decided also to go, so we thought, what if these are actually not astrocytes, but we are just mislabeling them as quiescent neural stem cells? So we went back to the whole dentate gyrus, so this is not sorted now. And what Alejandro tried to do was to map back those cells into the whole repertorium of astrocytes and neural stem cells in there to see whether there wasn't any specific subtype that then we could say, okay, so this subtype of astrocytes that was called astrocytes are actual stem cells. But he could not find anything like that. So basically they clustered all around this astrocyte slash neural stem cell cluster. So we were quite stuck. He tried to cluster, subcluster, sub-subcluster, and nothing was working until we actually found a paper of a new way of analyzing single cell sequencing data that really tries to maximize the amount of information of actual similarity that it maintains. So I really encourage you, if you're interested in this type of analysis, to check out this preprint. And actually, Dan de Groot, the first author, will be a group leader soon here at IMBA as well from the summer, so you should keep an eye on him. So we started to use his method and lately also to talk to him and get his input. And we analyzed, so I'm going to show you just exactly this same UMAP that was analyzed and done with Seurat, but in this case now with bonsai. So you can see it looks very different. It just gives you the similarity. So the more similar they are, the closer they will be. And all the first part were neurons. And then we focused on the non-neurons and started to want to see whether we can see some of these neural stem cells slash astrocytes in here. And using markers, we could identify 4 distinct clusters that express high levels of all these markers that will be common between astrocytes and neural stem cells. So, okay, then can now bonsai give us a separation between neural stem cells and astrocytes? And for this, what we decided to do was to map back these clusters to the spatial tissue, to the dentate gyrus. And we did this using available datasets from 10x, in which we can fully access. So we can just bioinformatically put back these signatures and see where will they land in the dentate gyrus. So exactly. So we did this. Alejandro did this. And he saw that astrocytes 1 to 3 had a very similar pattern in which they were all around as astrocytes will be, but cluster 4 was exclusively located in the supranuclear zone. So we think that we now finally have a way of really separating these neural stem cells from astrocytes, which means that Alejandro can finally go deeper and look at how heterogeneous they are and what hints of signals that can be regulating them can he find. And he's already finding some interesting novel markers that could be very helpful for the future. And one interesting thing that threads with everything that I've told you is that if we now look back at what was this cluster annotated as with the old methods, we see that some of these cells were annotated as stem cells, but almost half of them were previously annotated as astrocytes. So we think that with this new method of analysis, we are now recovering this big chunk of quiescent neural stem cells that we were considering astrocytes until now. Okay, so with this, I think I'm going to make a bit of a change of gears and I'm going to go to the last part of the lecture, which is what happens in the human brain. And here I'm again going back to old papers. So you can see this is before the 2000s, because this was one of the first evidence of some neurogenesis going on in the human dental jar. So they use a very similar approach as what I showed you for Alman and Dust in rodents. But in this case, they use BRDU. So it's not, it will be not radioactive, sorry. And these experiments were done because BRDU was given to some cancer patients and they gave their consent for their brains to be analyzed afterwards. And what they could see is that they could find some BRDU positive cells, mostly in the dentate gyrus, but also some in the subventricular zone. And this was really the first hint that something like adult neurogenesis could also be possible in the human brain. And the story of adult human neurogenesis has been and still is very controversial. So, for every paper demonstrating levels of adult neurogenesis, there's another one saying totally the opposite. And here's just one example. I think these are both from 2018, maybe. And you can see that in one case they say there's nothing, and the other one there's a lot. And I'm just going to go to show you the evidence that they showed. Yeah, this is the first one where they see that in adults there's barely any neurogenesis happening. So what they measure was DCX, which is double cortin, which will be labeling neural progenitors. So they will be already neuroblasts. And they saw plenty of them at birth and in very young kids, but from adolescents they could see pretty much nothing. So they concluded that adult neurogenesis was really not happening in humans, even though I still find quite interesting that the children do have quite a bit of it. But in a parallel paper, so almost at the same time, another group showed that they could find these double cortin positive cells in people from 20 to 80 years, and they could not even see such a strong decline in the number of neuroblasts as is seen in rodents. So they concluded the total opposite, which was that there was plenty of adult neurogenesis going on in humans and that it didn't have such a strong age coupled decline as it had in rodents. So you can see that the stainings, especially for this one, I would say they are not great. Double coating should have a much more like, yeah, different features, I would say, because it normally encircles the cell body and then has a very, very beautiful projection. That's what these neuroblasts have. And there's been a lot of back and forth with the technique because, of course, it's very difficult to work with adult human tissue because it's not very well preserved when it arrives to the lab. And this is something that groups like the one of Maria Llorens Martin have been working a lot lately. And they have developed a protocol, a very much improved protocol, in which now they can really get to work a lot of antibodies very consistently. And they have demonstrated already that these double cortin positive cells are indeed existing, and you can see that their immunos are much more convincing, I would say, than the ones of previous work. And they also have started to see differences between wild type, sorry, between control, let's say healthy individuals and those that have different diseases. And again, if you are interested, I really encourage you to look at some of this literature. But of course, immunos are not the only way of looking at this, especially lately. And in the past few years, there's been a lot of sequencing efforts also to see whether by single cell sequencing or single nucleus sequencing, we can identify these cells. And the story has been the same. So basically, you can see here in 2021, there was this first single nucleus, I think, sequencing of different species in parallel, including humans. And what they concluded was that they could not see anything similar to a radial glia, an IPC, or a neuroblast in the human brain. So, while they did see these cells in the mouse, in the pig, and in the macaque. So they concluded that there's really no adoneurogenesis going on in humans. But again, what happened was that there was another paper, also the same year, that used a slightly different technique for identification. So what they did was um, to really go looking for the neuroblast, and they used machine learning where they fed the profile of hippocampal neuroblasts during development in humans for it to learn how the transcriptional profile of these cells are. And then with that, they went to look for that profile in other data sets. So here you have their own data set, which they separated in different ages. And in red here, you can see where they find that this profile that they identified of young neuroblasts of the hippocampus was present or not. And they could see that this was present in all ages, including very old samples. And what was even more interesting is that they even used the data set of this previous group that I just told you that concluded that there was nothing, which will be this one, and other data sets that had also concluded the same. And even in those, they could see this profile popping up. Um, and what they concluded is that there were very consistent numbers of neuroblasts, but what they could not see were the proliferating cells. So they started to wonder whether human neural stem cells can enter quiescence, and maybe they were depleted. So this was quite controversial, and it's still quite controversial at the time. So in this comparison between mouse and human, let's say that what we can see is that there are these double quartin cells, but they are very low in number. So it's what they propose is that maybe double quartin is not the best marker. And this is a recurrent topic because many groups have seen that the markers that we consider good for mouse are not so good for human. And I'll go back to this a little bit with our own data. Um, but also, since by the time these proliferating precursors had not been found, one thing that was proposed is that maybe in the case of human, plasticity comes from a different phase. So it's not so much that there's a lot of stem cells waiting to be activated, but that a lot of neuroblasts have already been generated and are in what they call a plasticity phase where they don't fully mature, but only upon receiving the right signals, then they, let's say, reactivate from this stopped maturation and then fully integrate into the brain. But this is, again, as many things in this field, which is very exciting, still controversial, and we don't know. And with very new data, actually, we are now started to see that there are some of these stem cells in the human brain. And this is, I think, as far as I know, the latest one, which was published earlier this year, in which they already identified many neural stem cells, even some IPCs and neuroblasts in the human brain. And I'm just highlighting here another one, which is not so much centered in the neural stem cells, but I find it's quite interesting because they see also differences in the profile of the neuroblasts depending on the pathology of the individuals, and they focus on Alzheimer's disease. But also in this initial one, where they see a bit of everything, you can see that they have different groups, and some of those are what they call superagers. So they have also this AD, which is Alzheimer's disease. And you can see that the stem cells, which are here, so these are the red cluster, are more or less similar. So there are some differences, but at the level of the neuroblast, there starts to be quite that difference depending on the type of group that they look. So Alzheimer's disease versus these superagers, or young versus old. And again, please go to the papers for more details on this. And yeah, so going back to this identification of neural stem cells, there was also another paper recently where they really went to look for these proliferating precursors in here. So they used a very similar method as what I showed you for the neuroblasts. But in this case, they used the machine learning for proliferating precursors, so neural stem cells and intermediate progenitor cells that were proliferating. And then not only they did that, but also with the human samples from adults, they enriched the proportion of proliferating cells by doing FACS sorting. I think it was for Ki67, so really proliferating cells. And in this case, then they could retrieve a lot of neural stem cells from these samples, but of course they have enriched for them. But what they did not report at all, and I don't think they even mentioned, is quiescent neural stem cells. So they saw these active cells, they saw the progeny of these active cells, but they didn't even try to look for quiescent stem cells. And this is just to show you that I think one of the main reasons is that they fall into this astro/neural stem cell cluster, meaning that again, for human, it's very, very difficult to distinguish between astrocytes and neural stem cells because they are very similar cell types. And this is something that we are in the lab also looking at, and we think that with this same bonsai technique, we might get closer to really make the distinction also in humans. But it also makes evident that it's very difficult to study these states in vivo from this in vivo data if we don't even have a benchmarking or like something that we know for sure is an astrocyte or we know for sure is a quiescent neural stem cell. And now, as a last bit, I want to show you some work in the lab that we're doing to try to get an in vitro model for this. And this is something that Katerina in the lab has been doing for a while. She recently graduated. So as I told you, we have this very beautiful system in mouse in which we can isolate the neural stem cells and maintain them in vitro. And then with this BMP4 trick, they enter this quiescent state. So what Kate asked was whether this will also be possible in human. And she focused on seeing whether human cells which were exposed to this BMP4 and then also had a reversal, so a removal of BMP4, whether they will show these main characteristics or main hallmarks of quiescence, which is decreased proliferation, increasing glial features, and importantly, this reversibility, which will distinguish them from senescent or terminally differentiating cells. So we started this in collaboration with a lab in Innsbruck, which had isolated fetal cells, which we thought would be the best, so the closest to the real thing, and then maintain them in either neuroepithelial cell state, which is this highly proliferative, strong symmetric division state, which is not supposed to acquire quiescence, but also as radial glia, which is the state at which at least in mouse, we know these cells should be competent to acquire quiescence. And what she tested was these hallmarks, and she saw that neuroepithelial cells, as we expected, didn't check for any of them. Actually, they even proliferated more when we added the BMP, so the quiescence media to them. But the radial glia, even though they drop proliferation and increase GFAP, this was not reversible. And then we started to wonder whether this might be because not everywhere in the brain you can have these neural stem cells acquire quiescence. So we tested the fetal cells, these radial glia coming from fetal cells, and they actually did not come from a region that was even close to either the subventricular zone or the dentate gyrus. So what we decided to do is to use, so they were not patterned to the niche. So what Kate started to do was to use iPSCs and differentiating them to radial glia that were either not patterned to the niche or were patterned to one of the niches. And for the subventricular or subependymal zone, she collaborated with the Noblich lab here at IMBA as well. So when she tested the ones that were not patterned, she saw that they didn't they didn't acquire these hallmarks of quiescence, but the ones of the niches did, and that was really very encouraging. For the subependymal zone, there's a little bit more of variability, so we focus on the dentate gyrus, and then with this, we can now see whether we can extract a quiescent signature that we could then use to look for these cells in vivo. So just to show you how this looks, this is bulk sequencing of different control and BMP treated, so quiescent-like cells. You can see that they cluster very, very separately. So BMP does a lot to them. And what it does is very similar to what we observe in the mouse. So it increases lysosomal and autophagic functions. It increases the ability of the cells to process and listen to signaling cues. It decreases the cell cycle, and it also decreases metabolic features such as cholesterol biosynthesis. And what was very encouraging is that not only these three hallmarks, but also the whole transcriptomic signature, when we remove this quiescence media, really disappears. So they go back fully to what they were before, meaning that this is a fully reversible state. So we were very, very happy with this. And one thing that we then did was to compare not the general Go terms and what was changing, but the actual genes that were changing between mouse and human when we added this BMP. And we observed to quite a surprise that the genes were very different despite the processes being very similar. And we thought that this might be behind the difficulties in finding these cells in vivo again, because we were using, of course, the markers we know for mouse to look for these quiescent cells, and they might not be the same ones. So some things that we found were common are things like GFAP or CRY-IV, actually, that it's a marker that it's both increased in mouse and human neural stem cells when we add BMP, but has not been very used in mouse to label these cells. So this is something that we are following up, and I'll show you some images. And also we found some novel markers that were increased in these quiescent conditions in human, but not in mouse. And what we are doing now is to try to use this information to go back to this new data that we have from humans coming from sequencing from these two latest works. In this case, is the one where they have everything. So we identify the neural stem cell astrocyte cluster, and you can see that things like HOPEX, which is a general astrocyte slash neural stem cell marker, is a little bit everywhere. And same thing when we use the other data set, in which I'm here showing you the neural stem cells. So this will be just neurogenic lineage from the other paper. And again, it's very general, so it seems to be a bit expressed everywhere. And things like this marker that we identified, which is this CRY-IV, which is a marker of truncated radial glia. This also looks very general in this cluster, even outside the cluster. Same thing here. So it seems that it's marking something, but it's definitely not specific to the neural stem cells. But then the new markers that we have identified, like this SAMD11, now we start to see that it's not everywhere, but it marks a specific subset within this neural stem cell cluster, both in this data set and also in this data set. And we have some other markers, so this is another example that are even more restrictive, as you can see in both. But what is more exciting, and we are now doing in collaboration with a lab at the medical university, is that we can now look at human brain tissue. So this is a 10-year-old, and this is this CRY-IV that I just told you about. We still don't have the data for the unique markers, but we're really looking forward to that. Let's fingers crossed it will work. And here you can see that there's a beautiful staining. So the structure of the dentate gyrus is a little bit different, but I think you can recognize maybe this is the illus, this is the subgranular zone, and this is the granule cell layer where all the neurons are. So our stem cells, if the structure is the same as is in the mouse, should be located somewhere around here. And we see some of these CRY-IV positive cells everywhere. And I highlighted this one, which looks like glia. So we're not sure if it's an astrocyte, an oligodendrocyte, but definitely doesn't have a radial glia-like morphology. But there are some others, like this beautiful one here, that really does look like glia. So this fits very well with what we see in the sequencing, which is the cry-IB is expressed a bit everywhere. But when we have the resolution of seeing this spatially, then we can see that it marks these very beautifully distinct types of neural stem cells, one of which we think might be neural stem cells. So we are very excited about this. And with this, I'm already reaching my take-home messages. And I want you to remember that adult neural stem cells can generate neurons that contribute to brain function, that this is not something that happens in all the brain, but in specific niches, and they have specific functions. Actually, it's not really regenerating or some sort of homeostatic mechanism, but these new cells contribute a special function and a special addition to plasticity to these circuits. So this is a little bit different to what happens in other stem cell systems in the adult. Also, I mean, this is now going to come a little bit crazy, but that heterogeneity comes at multiple levels, and that is crucial for the function of neural stem cells because it allows them to respond to many different signals, but also to don't deplete their pool. And also that recent evidence is suggesting that adult human brain contains functional neural stem cells, and we definitely need new tools to study neurogenesis. I think it's a very active field right now in which single cell transcriptomics, proteomics, and hopefully soon metabolomics and signaling will be integrated to be able to also probe this heterogeneity and know more about the regulation of these cells. We need more in vitro models, and we need also in vivo biomarkers so that we can identify levels of neurogenesis in humans. Sorry. And yeah, what I was missing here because it didn't come at the right time is that neural stem cells are mostly quiescent, and this quiescence is important and very tightly regulated. And with this, I hope this was interesting to you, and it brings me to my acknowledgements, which will be my whole lab, which is fantastic, our many collaborators, and our funding. And I really hope you found this interesting. And yeah, good luck in the exam. And if you have any questions, you know where I am. Bye bye.